Hey there, welcome to chapter 34, Plant Form and Function. So in the next couple of chapters on plants and then animals, I'm not going to be covering every part of the chapter, and the parts of the chapter that I don't talk about in lecture will not appear on any exams. But you can feel free to read them if you're interested. Alright, so our objectives in this chapter is that you should be able to compare surface area to volume relationships amongst different shapes and explain the significance of these relationships. You should be able to describe examples of morphological diversity and phenotypic plasticity in plant roots and plant shoots. You should be able to explain the functional significance of the dermal, ground, and vascular tissue systems. You should be able to describe the role of apical meristems in primary growth. You should be able to explain the role of lateral meristems in secondary growth. Okay, so note that any chapters that are also being covered in lab will have this little sign here, so a lot of what we're going to be talking about we're also covering in Lab 9, L9. Okay, so the plant body is composed of a root system and a shoot system. So the root system is the part that's below the ground. It is the anchor for the plant. It takes in water and nutrients from the soil. Some of the nutrients that are really important to plants include nitrogen, phosphorus, potassium, and magnesium. The shoot system is the part of the plant that's above the ground. And so the goal of the shoot system is to harvest light and carbon dioxide from the atmosphere and then use photosynthesis to make sugars. So the combination of the shoot system and the root system make up the plant body. Alright, so the goal of roots and shoots is to absorb stuff. So the roots are absorbing water and nutrients, and then the shoots are absorbing light. And so the absorption takes place across the surface of the plant, and then the product of the absorption has to take place inside the plant, right? So a plant is going to be very efficient in its absorption if it has a large surface area relative to its volume. So it has a lot of outside compared to how much inside it has. So if we kind of make a model here where each cell, we'll imagine a cell being like 50 nanometers across. I'm sorry, that's a micrometer. 50 micrometers across, so 50 by 50. And we'll say if you stack them all up, the shortest, squattest shape you can make is a cube, right, of 64 cells. And that would be like a shape that would be analogous to something like a potato, where it's like as short and squat as it could possibly be. And so if you add up, the surface area, 50 times 50, and then you just, 50 times 50 times 96 cell surfaces, all right? Your final cell, your final ratio, So your final volume, if you look at the volume of each of these little cubes, is going to be 8 million micrometers squared, and so then the ratio of your surface area to your volume is going to be 0.3 micrometers, okay? All right. If you take a structure that is kind of like a root, and to replicate a root, we've taken all of our little cubes and put them in a long tube like this. Or it's not a tube, it's solid, but you know what I mean. It's long and skinny. All right, so then if we add up the surface area, of the 50 times 50, plus 50 times 50, etc., all the way around, all four sides, we get more surface area compared to this kind of a squat shape. So we've gone from 240,000 micrometers squared to 340,000 micrometers squared. The volume is going to stay the same because we got the same number of cubes, right? So it's still 8 million. So our ratio of surface area to volume has increased to 0.0425, okay? If we look at something like a leaf is basically like a flat, kind of a structure. So if we took all of our cells, our cubes, which represent cells, and laid them flat like this, we've now increased the surface area to 420,000 micrometers squared, and then the volume is 8,000. So what does that do to the ratio of surface area to volume?
It should be bigger.
Yeah, that's pretty good, dog. I didn't think you knew how to do math.
I don't. I'm good at following patterns.
Okay, you are correct. So the surface area to volume ratio of a flat leaf should be even greater. Okay? All right. Let's look at the root system. So the root system of a plant does a lot of different things. It anchors the plant in the soil, obviously, so the plant doesn't wash away. Its job is to absorb ions and water from the soil, and then take the water and ions and conduct them to the shoot system, so get them out from under the ground and bring them up above the ground. It needs to be able to get the energy from the sugar that's being made in the shoot system, and it might need to store molecules produced in the shoot system. So plants that, make, I don't know, like tubers or really thick tap roots, like your potatoes or carrots, those are basically storage systems for extra starch to be used later. So it may need to do that. There's a lot of diversity in root systems. So you can see here, this is like just like the lawn that you might have outside of your house. It has very shallow roots, and you know that it does because you can just peel it up if you need to. Something like a lead plant has really, really deep roots. Pale purple coneflower or June grass or switchgrass. Compass, oh, that was June grass. This is compass plant. These are kinds of prairie plants that are native, and they tend to have longer, deeper roots. All right, so what I just showed you, the previous slide where we looked at diversity amongst roots, that was diversity that varied from species to species, right? There's also phenotypic plasticity in roots. So phenotypic plasticity refers to the ability of one genotype to produce more than one phenotype when exposed to different environments. So, for example, phenotypic plasticity is related to acclimatization. So, Dog, what would your fur look like if you sat outside all winter?
Oh, it would be nice and luxurious.
All right. What would it look like if you stayed inside all winter?
Just regular.
Okay. So the dog has the same genotype, but he has different phenotypes depending on his environment. Same deal with plants. So the environment will affect the type and the depth of roots. So you can take one plant that's like a clipping of another plant, so they're genetically identical, and grow them in two different places, and they'll often end up with different kinds of roots. So roots will grow in the area where there's stuff, like resources, ions, and water, and then the roots will die back in areas without the resources. And so plants have a variety of adaptations that they can use to adjust to the environment. So some plants will form anchor roots, and so these are also, this kind of anchor root is an adventitious root, meaning it comes up when it's needed to cling to a surface. So you get that in ivies sometimes. So if the vine is right next to a tree or something, it'll grow these little roots. You can get prop roots. So if you have a plant that is growing up out of the ground like this, and it looks like as it gets bigger and bigger, it gets more and more unstable, it'll grow prop roots to support it. Have you guys seen any of these prop roots on any plants we have around here?
Yeah, I've seen that for sure. That's like how the corn is on the bottom of the corn.
Yeah, do you go into the cornfields?
Well, yeah, I'm a bird. Go in there to eat stuff.
Yeah, so corn has this. You have structures called nematophores, and so these are these little structures that come up from the roots if the plant is waterlogged sometimes, and these will gather oxygen, or sorry, gather carbon dioxide. And then you also have storage roots like this, like, a radish or a carrot or something, or a sugar beet, which is used to store carbohydrates. And if you, have a really good season, there'll be really big fat sugar beets, and a really, kind of weak season, they won't be large at all. Okay, so when comparing the root systems of corn plants growing on a square foot in a fertilized and irrigated cornfield with the root systems of natural prairie plants growing in a meadow, what result do you expect to see?
I don't know.
Well, if you had to guess.
Why don't you ask the bird? She's eating more corn. Yeah, okay. Well, I guess I would guess that if you give them plants fertilizer, then they probably wouldn't need to grow very big roots. And if you had no fertilizer and no water, they probably have to grow really big roots.
That sounds reasonable. Let's see what the choices are. The overall mass of roots will be identical in a cornfield and on a meadow.
No.
The mass and length of roots in a cornfield will be higher because the soil is fertilized.
No.
The mass and length of roots in the meadow will be higher because the soil is poorer and there is higher diversity of plants.
Yeah, I like that one. Maybe like the diversity will make it so the plants will have to compete.
Yeah, that's true. The overall mass of roots in a meadow will be lower, but the length will be higher since the roots need to grow deeper to reach nutrients.
Why would the mass be lower?
Well, that's a good question. I don't think there's a good reason why the mass would be lower. Okay, so let's look at the shoot system. So again, this is the same picture, but with more words on it. So the stem is the part above the ground, and the leaves are attached at, the places where the leaves are attached, we refer to that as a node. So this is a node, and that's a node, and that's a node, okay? And then you could say the sections between the nodes are the internodes. And these could be really long or really short. Okay, so there's two kinds of buds. There's axillary or lateral buds, and they form at the nodes above where the leaves attach, so kind of in the armpit of the leaf, right here. There's another one. So an axillary bud forms here and here, and here and here. And then the axillary bud may grow into a branch. Apical buds, right, that comes from the word like apex, so that's the one at the top, okay? And the apical and the axillary buds may develop into flowers depending on the kind of plant. All right, so there is natural selection acting on plants all the time, and some plants have managed to get lucky mutations that allow them to survive unusual circumstances. And so some of these adaptations have happened in parts of the stem structure. So you have modified stems like a cactus. This puffy part of the cactus is part of the stem. The spines are actually modified leaves, which is the next section we're going to talk about, but this big thick part is the stem. Stolons, like strawberries have, those are modified stems that allow them to reproduce asexually. Rhizomes, right here, is part of the stem that is below ground and it stores carbohydrates. Tubers, like potatoes, those are also carbohydrate storage, and those are also modified stems. Thorns are a modification of the stem to protect from herbivores. And there is a structure here called a corm, okay?
Was that corn?
No, not corn, corm.
Corn?
No, not corn, corm.
Oh, corm.
And so a corm looks a little bit like a bulb, but they're not the same on the inside. So it's actually kind of solid on the inside because it is a modified stem. So if you cut it open, you can see instead of it being layers of leaves, it's actually a kind of a swelling on the stem. All right, so there's a lot of diversity in leaves as well. So if you have a plant with a simple leaf, just looks like this, and it has a blade like this, and a petiole, which is the stalk. A compound leaf blade is divided into leaflets. So you have kind of one petiole with lots of little leaflets. And you can have a doubly compound leaf. So you have a compound leaf, then it splits again into more and more. You can also have things like needles. And this is an adaptation to minimize water loss if you live in a very cold or a very hot climate. And there can be variation in how the leaves are arranged on the plant. So they could, each node, the leaf could come out in an opposite direction. So those are alternate leaves. At each node, leaves could come out of both directions. So those are opposite leaves. You could end up with a whole bunch of leaves going in different directions at particular nodes. So those are whorled leaves. And then sometimes, if you were to take the distance between each of the nodes and shrink it way down, you would end up with a rosette like this. Okay, so there's also phenotypic plasticity in leaves. So we know that just even in the same tree, there are sun leaves and shade leaves. So the sun leaves, they're in the sun, right? And so it is advantageous for them to grow in such a way that they have a very small surface area so that they don't lose too much water, since they can get as much light as they need, versus the leaves that are in the shade. So they're relatively large and broad because they need to have a high surface area to get enough light to be able to photosynthesize. There's all sorts of adaptations to modify leaves as well. So this is a bulb. So if you cut an onion, right, each of these is a leaf. You know, when you peel off the layers of an onion, those are each modified leaves. And that was achieved by shortening the nodes, right? So all the nodes have been shortened so that the leaves are all stacked one on top of each other. So in some way, this is a leaf modification, but it's also a stem modification, right? Let's see, succulents, like this aloe plant. So the leaves will store water. Plants can have tendrils that come off of, that are modified leaves. There's also some tendrils that are potentially modified stems, but in the case of peas, these are modified leaves. you have floral mimics, so you have structures that look like petals, but they're actually colored leaves. You can have traps like this, like leaves that have been rolled into a little like drowning pit for insects. And something like a cactus, the spines will protect the stem, and so the spines are actually modified leaves. Okay, so as you know from 1113, plant cells have many features that are not found in animal cells. So plant cells have a primary cell wall, which is made of cellulose. Some plants also have a secondary cell wall, and there are little holes that connect one cell to another so that the cells can communicate with each other. So the holes are called plasmodesmata. Okay, remember that? Plasmodesmata? And you can even see here that the rough endoplasmic reticulum, the smooth endoplasmic reticulum is kind of leaking between the two cells and communicating materials through from one side of the cell to the other. All right, so some botanists make the argument that the entire plant should be considered as a single unit rather than a composite of many individual cells. Which of the following cellular structures best supports this view? Cell wall, cell membrane, cytosol, vacuole, and plasmodesmata.
Oh, it's the plasmodesmata because that's the stuff that's going back and forth between the cells like it's one organism.
Yeah. Yep.
What's cytosol?
That's like that kind of gooey stuff that is inside the cell. It's kind of a soupy goo, can be found within organelles.
Oh.
Okay. So plants have three tissue systems: the dermal tissue system, the ground tissue system, and the vascular tissue system. Within the ground tissue system, there's three flavors of ground tissue: parenchyma, collenchyma, and sclerenchyma. And then in the vascular tissue system, there's two kinds of tissues: xylem and phloem. All right, so we're going to start with the dermal tissue system. So the very outside of the dermal tissue system is the epidermal cells, and so those are the cells that secrete a waxy cuticle, and so that's what keeps the plants from losing water. Also, it protects them against invading pathogens that might viruses and bacteria also affect plants, and so the waxy cuticle can repel some of those. But plants need to be able to take in gases in order to photosynthesize. So there is in the dermal tissue stomata, right? So those are the holes, and their job is to regulate gas exchange and water loss. And so the holes are guarded by these two guard cells. So you can see that when the cells are full of water, they kind of puff up to be kind of bean-shaped. So here's one of them, and then here's the other one. But when there's not very much water, the holes kind of flap, the guard cells flap shut, and that closes the hole. Okay? And so that way, when the plant is starting to dry out, the guard cells will lose water, and then the stoma will close. Stoma singular, stomata plural. Then the plant won't lose any more water. But when there's plenty of water around, the guard cells will puff up and open up the stoma, and so then photosynthesis can happen. One component of the dermal system that you might not think about are the trichomes. So these are these little hair-like structures that are made up of epidermal cells, and so they can do all sorts of things. If you've ever seen a plant that's kind of fuzzy looking, the trichomes can reflect sunlight to make the plant cooler. So a lot of like desert plants will have little fuzz on them, like kalanchoe, for example, if you have one of those. It can reduce water loss by limiting transpiration. So as water is leaving the plant, the kind of layer of hair holds onto the water so that there's a moist environment where the stoma are. Sometimes there's barbs or toxins to kind of get herbivores. And in cases like a honeydew plant, there's sticky stuff in them to trap and digest insects. All right, so the next layer, or the next kind of tissue is the ground tissue. And remember, ground tissue has three types of tissue within it: parenchyma, sclerenchyma, and collenchyma. Okay?
They just rhyme. It's wonderful.
Yeah? You like that they rhyme?
It's like Huey, Louie, and Dewey.
Okay. Are those ducks?
Yeah.
All right, so parenchyma is what is known as a simple tissue, meaning that if you look at it, it's all made up of a single kind of cell. And parenchyma cells have pretty thin walls, and they are the most abundant and versatile plant cell. So most cells are parenchyma. If you look at the leaves of a plant, the parenchyma tissue is where photosynthesis takes place, and in the roots, parenchyma cells are where you're storing starch, and so we've stained these starch molecules purple, so you can see that they're stored within the root cells. You can see these are parenchyma cells because they have really thin outlines on them. These are parenchyma cells that are photosynthetic, because you can see they have chloroplasts in them, and they also have a very thin cell wall on the outside. All right, the next kind of ground tissue is parenchyma. It's also a simple tissue. What's that mean if it's a simple tissue?
One kind of cells.
Yeah, one kind of cells. And its job is to provide support to the shoots above the ground. And if you look at parenchyma up close, they look kind of weird because they have unevenly thick primary cell walls. So you can see they're kind of weird shapes. And so parenchyma cells are providing flexible support as the plant is actually growing. So you often find them just outside of the epidermis of the stem, especially outside of vascular bundles. So this is a celery stalk. You know how it has those strings in it? This is one of those strings, which is made up of parenchyma. If you look below that, right, that's the vascular bundle. So that's where the water is being carried. All right, the third kind of ground tissue is called sclerenchyma. And sclerenchyma is made up of two cells, okay? So let's see. Yeah, so they have a thin primary cell wall and a thick, rigid secondary cell wall. And so this really thick cell wall contains not only cellulose, but also lignin. It's a complex tissue because it's made up of two kinds of cells, like I just said, fibers and sclerids. So the fibers are long and skinny. I mean, if you see them in cross-section, they just look like little cells, right? But just keep in mind, that's because they've been cut like that. And then the sclerids are short little cells that are more variably shaped. All right. One major difference that you would find between the sclerenchyma and parenchyma cells is that the sclerenchyma cells are dead at maturity. So when they actually are ready to do their job, they are basically dead. So they grow until they get to their final size, and then they die, and only after they die is their job starting. So their job is to provide support. Parenchyma cells, in contrast, have expandable primary cell walls, and they're alive at maturity. So parenchyma cells are still growing. The sclerenchyma cells are doing their supporting work after the cell has died. All right, let's compare and contrast parenchyma, parenchyma, and sclerenchyma. So how do they differ in number of cell types?
Well, the parenchyma and the parenchyma only have one type of cell, but a sclerenchyma has two.
Yeah, that's true. Remember the two kinds?
Like fibers and sclerids.
You want to look again?
Yeah.
Yep, fibers and sclerids, right there. So how do parenchyma, parenchyma, and sclerenchyma differ in function?
Well, like the parenchyma is just used for lots of things, and the parenchyma is like flexible support when it's growing, and the sclerenchyma is like really strong support when it's mature.
Yeah. How do they differ in cell wall structure?
Oh, well, the parenchyma's got real skinny cell walls, and the parenchyma has like uneven cell walls, and sclerenchyma has like reinforced cell walls with lignin in them.
Yeah. All right, so alive or dead at maturity?
Oh, the parenchyma and the parenchyma are alive, but the sclerenchyma are dead.
Yep. All right, now let's move on to the vascular tissue. Okay, so we have two kinds of vascular tissue, xylem and phloem. Xylem is the tissue that's used to conduct water and dissolve nutrients from the roots up to the shoots. And so there are tracheids, right? So these are long tapered cells that have pits in them to allow the fluid to go up, and this is what they look like. There are thicker ones called vessel elements. And so these are short and wide, and they have much bigger perforations and pits to be able to exchange water from one cell to another. And you can find tracheids and vessel elements together in vascular tissue. So you can see these little skinny, kind of browner ones are the tracheids, and then these really big ones here are the vessel elements. Okay. The phloem, the job of the phloem is to conduct the sugar and amino acids and hormones from the roots to the shoots, but also from the shoots to the roots. So the xylem is moving water up from the ground, but the phloem is moving water all over. So don't get confused and think xylem moves water up and phloem moves water down. Phloem moves water down, but also phloem moves water up and side to side. So phloem is a complex tissue. Okay, so there's two types of cells called sieve tube elements. And so these are these long, thin cells. And then at the end, they have something called a sieve plate. Do you know what a sieve is?
Is that what you stab somebody with in jail?
No, I think that's a shiv. Okay. No, a sieve is something different. Anyone know what a sieve is?
Yeah, it's like that thing in the kitchen with the holes in it. You can, like, use it to, like make your pasta.
Yeah, kind of. It's like a colander. You know, you might use it to, like, pan for gold or something like that. Okay? So sieve tube elements have sieve plates at the end, so there's little holes in them. Sieve tube elements have no nuclei, and so they directly connect to a companion cell by plasmodesmata. So the sieve tube elements are kind of real dumb cells with no nucleus, but they have a companion cell on the side. So here's a sieve tube cell. You can see that it is because it's got a sieve plate showing, and then it has a little companion cell with a nucleus. And so the companion cell is kind of the cell that's controlling what's going on, and it is connected to the sieve tube element through plasmodesmata. Okay, so we have two kinds of growth. We have primary growth, which is basically making things taller, okay? So making the shoots taller and making the roots deeper.
Wow, that was a very sad story.
Well, look, it's starting again. See?
Ah!
Okay, so secondary growth is the kind of growth that makes plants fatter. So it increases the width of roots and shoots, and it increases like the kind of vascular tissue that allows liquids to be moved around throughout the plant, and also when that happens, it gives more structural support. So secondary growth is what creates wood, and it occurs in species that have a cambium, which we'll talk more about in lab. All right, so again, the primary growth, do you remember which direction is primary growth?
It's up and down.
Yeah, up and down. And so you can see the root, the primary growth is happening right here, right? In the apical meristome of the root. And then in the shoot, you can see the apical meristome is up at the top. And so the meristomes will allow plants to grow taller and taller. Excuse me. So these cells are undifferentiated, and they always have the ability to undergo mitosis and continue growth for the plant's lifespan. Yeah. So apical meristomes are responsible for primary growth. And so the apical meristome will give rise to three different kinds of meristomes. So there's a kind of a meristome, or sorry, the kind of meristome called protoderm is what's going to give rise to the dermal tissue system, and ground meristome gives rise to the ground tissue system, and procambium gives rise to the vascular tissue system. This is a little bit like what we learned about animal development, didn't we? Didn't we learn about different layers?
Oh yeah, like endoderm and ectoderm and mesoderm, giving rise to the different systems of the body.
Yeah, it's a little bit like that with plants, right? Okay. And here's the roots. So the root apical meristome is protected by a root cap. So the root cap is not only protective, but it also can detect gravity to determine which direction the root should grow in. It also secretes slime to make less friction as the root pushes its way through the soil. And then beyond the root cap, there are three zones of cells. So you have the zone of cellular division, which is right behind the area where mitosis is happening, or right in the area where mitosis is happening. you have the zone of cellular elongation, where you can see that the cells start to lengthen. And then you have a zone of cellular maturation higher up, where the cells are differentiating into either dermal, vascular, or ground tissue, and so you start to see things like root hairs sticking out, and these can start to be able to absorb water and nutrients. All right, so let's take a look at the primary shoot system. In a eudicot, vascular bundles are arranged in a ring near the stem's perimeter, meaning near the outside. So you can see here, each of these is a vascular bundle, like this. And the ground tissue is divided into a central pith and an outer cortex. So this bit on the outside is the cortex, and this bit in the middle is the pith. If you're a monocot, your vascular bundles aren't arranged all around the outside, but are instead scattered throughout the ground tissue. Oh yeah, and they kind of look like screaming monkey faces, don't they?
No.
I mean, I think they do. Okay. So secondary growth, meaning, do you remember what kind of growth is secondary growth?
It means the growth to make the plant fatter.
Yeah. So secondary growth is increasing the width of the plant, and so this is something that happens yearly. There's a layer here called the cork cambium, and so it's a ring of cells right near the outside, and it produces cork cells on the outside of the trunk, and so that kind of gives rise to the bark. There is the vascular cambium right here, and it's a ring of cells between the secondary xylem and the secondary phloem. So this bit here in red, so these are rays of phloem here, and then these are rays of, and these are where the xylem is over here. So this is showing you the vascular cambium here. All right, so the vascular cambium produces secondary xylem, which is wood, basically, and secondary phloem, which becomes the inner part of the bark. So where's the primary xylem and phloem?
Oh, it's in the middle.
Yeah, it's way in the middle, right? It's very in the very inside part. All right, so if you look into a tree trunk, you can see that it has like a dark part and a light part. So the heartwood is the dark inner xylem, right? That's the part that's conducting water up from the bottom. Sapwood is the light outer xylem. And if you have an area in which you have like a hot season and a cold season, the vascular cambium will stop growing when it's too dry or too cold. And so you end up with a structure of annual rings every year. All right, so where are the cells that cause primary growth?
They're in the middle.
Yeah, well, I guess they're in the middle, although where are they when the plant is young?
Oh, at the top and the bottom.
Yeah, they're the top and the bottom. So what are they called?
The top one's the apical meristem, and the bottom one is the root meristem.
Yeah. So where are the cells that cause secondary growth?
Oh, they're like, there's a row at the outside, and it's something cambium, and then there's a row between the, I don't know, and it's called something cambium.
You want to look at it?
Yes.
Okay. So we have the cork cambium right at the outside here, and then we have the vascular cambium that gives rise to the xylem and the phloem over here. Okay?
All right.
All right, so hopefully in this chapter you learned about roots, shoots, and leaves. You learned about variation in plant form, the three kinds of plant tissue, how plants grow primarily, and how plants grow secondarily.